His work focused on multivariate data analysis and explored orientation invariance in the human visual system using fMRI. As a postdoctoral researcher, he continued to contribute significantly, studying topics like human learning of abstract categories and transfer learning. Hamed Nili currently conducts research at the Nuffield Department of Clinical Neurosciences. His expertise lies in analyzing the information content of multivariate neural activities. Dr. Nili, thank you for accepting our invitation. We are thrilled to listen to your talk.
بهم بگید که اینا این لیزرش رو perfect. Okay, thank you very much. It's my pleasure to be here and to talk to you about my recent research. First, I thought the talk is going to be in Farsi, so I thought about the words that I'm going to be using to translate these into Farsi, but now I realize that I have to speak in English. So it's easier from one perspective, more difficult from another perspective. Today, I'll be talking about mapping, encoding, and flow of task-relevant information from human MEG recordings in a perceptual decision-making task. And so before anything, I'd like to acknowledge all of my colleagues who contributed to this research. Marco Cellotto is a PhD student in Stefano Panzeri's lab. Alessandro Tosta is a postdoc in Tobias Donner's lab. And Toby and Stefano have also been part of this project. Okay, so what is perceptual decision-making? And perceptual decision-making is the process by which animals detect, discriminate, and categorize information that comes from the senses. So, for example, I get some visual input, I process it, I do some processing on it, and then I make a decision of, for example, let's say like whether this bar is tilted to the left or to the right. And so it requires encoding of sensory input and generating appropriate behavioral choices. We see the brain as an information processing system, and it sounds very reasonable that we take the information theory framework as a tool to study brain function. So as an example, I'd like to point out to this particular design, which I'm going to get to a lot during my talk. This is an example of a visual discrimination task. The subjects at the beginning of, so this is what a single trial would look like. The subject at the beginning, they see a reference grating with a fixed contrast level, let's say 0.5. And that is followed by 10 successive samples, one presented one after the other. And then they have to make a decision, and they have to report their decision whether they think the average contrast of these 10 gratings is weaker or stronger than that of the reference. So it's a visual discrimination task because they have to discriminate whether the average is larger or stronger, and they have to accumulate evidence across these 10 samples. And they respond with two hands. So, for example, if they think the average contrast is stronger, they respond with the right hand, and if they think it's weaker, they respond with the left hand. Sorry, this is okay. And the objective here, like most of the other neuroscience research, is that we would like to use information theory on whole brain measurements. And the idea is that we would like to ideally reverse engineer the brain function and see how does the brain do it. And then if we understand that, we could probably transfer this to machines, etc., etc. So in this particular experiment, we are measuring brain activity via MEG recordings. And I'd like to spend like 1 minute explaining what MEG is and why we think this particular data set is useful for us. So MEG measures the neural activity through the magnetic field that is generated by the neurons. And it allows whole brain recording, so we can measure the whole brain altogether with a very good temporal resolution. The idea is that we have temporal resolution of the order of, let's say, 1 millisecond. However, the downside is that the spatial resolution is pretty poor. So because what we measure is the signal on the skull, right? and then from that we have to solve the inverse problem and infer what the activation is at the cortex, for example. Because of this inverse problem, it has some caveats. The spatial resolution is pretty poor. Is relatively poor compared to fMRI, which is very precise in space. But the temporal resolution is very good. Now, in this task, we are using a pretty sort of like reliable way of estimating of sort of like source localization, and we're confident that the signal that we have has a relatively good spatial resolution. We have data from 15 subjects, and each subject have, let's say, in the order of 400 something trials. So it's pretty much like the electrophysiology studies where they have many, many trials. And this makes it particularly useful for information theory, because with information theory, we need a lot of trials. It requires a lot of data, and we have that data with this particular measurement and with this particular setup, if you like. So then what we do is that we infer the activity at each cortical vertex, they call it. It's like it's the smallest response channel, let's say, at the level of the cortex. And then what we do is that we average across the vertices that belong to the same, they call it parcel or region of interest, and we have one sort of like data per region of interest or parcel. And this is a very sort of like very well-known atlas, which was proposed by Glasser and colleagues. It's a multimodal parcellation. The idea is that they divide the cortex into 180 parcels or regions of interest in the right and the left hemisphere. So in total, we have got 360 ROIs, and we can use that data we can use the data from different ROIs to see what's going on at the different regions. The reason that I'm averaging the data across the different vertices that belong to the same ROI is that I gain some SNR, because I'm pooling across the vertices that have almost similar properties. I reduce the effect of noise by doing that. Okay, so a bit about information theory. This will be very short. So imagine that I've got this stimulus, in our case, for example, the contrast of the gratings, and I'm measuring the response from this region in the brain. This is the response across trials, right? So if I have S for many, many trials, and I have got R for many, many trials, what I could do is that I could compute the mutual information, which I'm sure all of you are familiar with, between the stimulus and the response. This tells me in an assumption-free way and at a single trial level how much information we get about the S by looking at the R. So it tells us about the stimulus information and about the encoding of the sensory input, which is one important aspect of the perceptual decision making. And then we can look at the mutual information between the choice and the response of the region. And this tells us about the behavioral readout or the choice information. So it's about the other side of the process. Now, importantly, we can also look at with the recent developments in methodology, we can also look at the intersection between the sensory encoding and the behavioral readout. So this measure is called intersection information, and this tells us how much of the sensory input in one region is informed to form the choice. So basically, like I've got this much bits of information in this region, how much of that is part of the choice information? And just to sort of like to explain that a little bit, okay, I don't know how to get rid of this, but fine. So it's the minimum of two components. One is the part of the stimulus information that is about the choice, and the other part is the part of the choice information that is about the stimulus. These are called partial information decomposition atoms. The algorithms works like this, that we get this one, and then we get the minimum of the two, and that will be the intersection information. So the idea is that the regions that contain intersection information have the neural code that is essential for doing the task. So it's very important that we sort of like look at this measure, if you like, for the different regions. Yeah. اینو من نمیدونم. آهایدو بزنین. اوکی. حالا بزنین این کینوت این یکیه. Okay, merci. Yeah. Now we can also, with information theory, we can also look at the information transfer and how the different regions communicate with each other. So for example, if I've got region 1 and I've got responses from region 2, then we can look at the information propagation by looking at by measuring transfer entropy. I will explain this in a couple of slides. And then we can, with the latest tool that we've recently developed, we can also say what aspect of the information is being transferred from one region to another. So for example, we can compute the feature specific information transfer about the stimulus. The idea here is that region 1 is sending information about the stimulus to region 2. Because if you know, with transfer entropy, we're blind to the content of the transformation. Here we're looking at the context as well. And sorry, one slide back. And we can also do the same for choice and compute FIT about the choice. Okay, so I've explained the task already to you. You know what the task looks like. It's one reference, 10 samples, one after the other, and then the subject makes the choice. They report the choice. And I'm looking at the data from these different regions of interest in the brain. Right, so here we wanted to take a very exploratory approach. Do not make a priori assumptions about how and where the information is located. So what we do is that for each of the regions in the brain, we compute the stimulus, choice, and intersection information as a function of frequency and time. So we have got these. We have for each pair of time and frequency, we have got data from many trials in each region, and then we can compute the stimulus information matrix, the choice information matrix, and the intersection of the two. And then we can see how the data is spatially and temporally distributed. So, and the neural data is always aligned to the onset of the first sample. So here we align all of the trials with respect to the presentation of the first sample. Let's look at some results. So here I'm showing you results from V1 and M1. V1 is known to be the first part of the visual processing hierarchy. We expect V1 to contain stimulus information. So when I look at the stimulus, and here's what I get. When I look at the stimulus information in V1, the time frequency, and what I show is that I show the significant parts in colored and the non-significant parts are in gray. So what we see is that from the onset of the stimulus up to the end of the presentation of the stimulus, there is significant stimulus information which is localized in a particularly narrow frequency band. So this is the gamma band for those who are familiar with it. So we have a significant stimulus information in the gamma band. This is expected. We expected to see this, and it's like a proof of concept, if you like. And then we go to the other extreme of the visual hierarchy, of the processing hierarchy, which is M1. That's where, because the subjects, they respond with the hand, so we expect to get choice signal in M1. And what we see is that we have significant choice information, which starts being significant from 0.5 onwards, and is pretty much in the lower frequencies compared to this gamma. So this is alpha plus beta, and this is theta. And next we can look at these other sort of like variables of measures of interest. What we see here is interesting. What we see is that this stimulus information, which we saw in V1, is also part of the choice information and intersection information as well. So this means that V1 is not just reflecting the sensory input which is out there, but what the signal is in V1 is also choice related. And then very interestingly, what we see in M1, which is like known to be sort of like only responsible, let's say, for motor execution or for motor planning, is that we also see significant stimulus information appearing pretty early on, like say at around half a second after the onset of the first sample, to be significant, and that is part of the intersection information as well. So this was very interesting for us. The fact that M1 contains the stimulus information and V1 contains choice information. And here are the results for some other regions in the visual processing, in the processing hierarchy. Here is V1. This is a region in the posterior cingulate. These are two parietal regions, and this is the M1 again. So so what we see is that if you look at these regions, we have got in most of these intermediate regions, we see this gamma stripe in the stimulus information, and we also see this alpha plus beta in the choice information. What I want to say here is that these regions have signatures that correspond to both of the extremes that we saw, the V1 and M1. And so then you remember at the beginning I told you that we have poor spatial resolutions. We might have poor spatial resolution with MEG, but we're using a sort of like a state-of-the-art source localization method, we're pretty confident that the signals that we have are localized in space as well. So what I said we could ask is that these regions in the brain, they belong to 21 different anatomical groups. And this is what the Glasser Atlas suggested. So we could, I told you we've got like 180 regions per hemisphere. We could divide them into 21 distinct anatomical groups. Let's say like the early visual processing, the motor cortex, I don't know, the posterior cingulate cortex, and so on and so forth. And then what I said is that we could see whether the information profiles, meaning the time frequency and matrices that we got for the different measures, whether they are decodable, whether we could decode the region, the group, if you look at the information profile. And here is what we see. So I trained different I used the support vector machine, and I trained different SVM models on the time frequency profiles and see how well they could predict the anatomical group that they belong to. So what we see is that here we've got 21 groups. Of course, the chance level is 100 divided by 21, which is 4.76. But we see that the performance of these different models that we get is at around 70%. So we're doing way, way better than the chance level. And then another interesting point is that if we use the intersection information as the feature, it is the one that provides the best discriminative set. So it means that these anatomical groups are mostly discriminative when we look at the intersection information. And here is the confusion matrix, just to validate what I just said. We see that the off diagonals are very small. We just see some confusion in the frontal areas, which makes sense because they're pretty close to each other. Okay, in this part, I'd like to identify the task relevant frequency bands. So we get these different time frequency profiles for the different regions, and then we can pull across time. We consider the stimulus presentation window, which is 0 to 1, and then we average across time, and we've got this information versus frequency for the different parts of the brain. And then what I'm showing you here is in color, the histogram of information versus frequency for the stimulus choice and intersection in different panels, and for the different anatomical groups. In this talk, I'm always going to show you results for the different anatomical groups because they sort of like provide a way of sorting the regions. And this is, the first ones are the auditory cortex, which is like the lowest in the hierarchy. And the last one would be the motor cortices. And just to explain how I get these is that this is based on, there was a Nature Neuro paper which suggested that, in fact, they showed that the T1 over T2 ratio provides a very good proxy to the anatomical hierarchy. So what I'm doing here is that I'm sorting these groups according to the T1 over T2 ratio, except from the motor areas that I bring to the very last, because in this task, they would be the last part of the processing. So very interesting what we see in the stimulus information. This is our gamma band that we saw earlier. And for choice, we can see that we have this strong alpha plus beta. And we also have this gamma, which was part of if you remember in the visual areas, the gamma band also contains significant choice information. And then when we look at the intersection information, we clearly see that there are two bumps in these results. This is the gamma, which is like 40 to 80 hertz, 40 to 75 hertz. And this is the alpha plus beta, which is 8 to 40 hertz. And then we also see this small theta, which I'm not considering in this presentation, and I will explain to you why. So we have got these two dominant frequency bands that come out of the results without making any assumptions about which bands we have here. So it's interesting that although we did not make any priori assumptions, the bands that we got sort of correspond to the ones that are well known in the literature, to the so-called brain rhythms. And here we can look at the spatiotemporal information profiles for the different, so this is the gamma rhythm, and this is the alpha plus beta band. This is stimulus information. And this is choice information. This is the intersection of the two. And this is what I call the pure stimulus information. This is like the part of the stimulus information which is not choice related. And this is what I call the pure choice information. So just looking at it immediately, what you can see is that there's a lot of stimulus information in the gamma band in these upper part of the matrices, which are the visual areas and the posterior cingulate and the parietal cortices. And then when we look at the the choice information in the alpha plus beta, they're pretty large in some of these groups that we see. So these are the motor cortex, the premotor cortex, the parietal cortices, mid-cingulate cortex, and the posterior cingulate. So, and then we see that the stimulus information is largest at the beginning, choice information is largest towards the end of the presentation, towards the end of the response period, and then we see that this stimulus information is also some of it is also not choice related, and some of this choice information is also not stimulus related. I just want to mention this point about this, that all of these that we could get, that we could decompose the information so nicely into the parts that are choice related, for example, the stimulus information that is choice related or that is independent or unique, or which is unique to choice. It all comes from the PID, or the partial information decomposition framework. It's very, very useful for our analysis. And just to recap what we got. So for example, this is the V1 in the gamma frequency. As you can see, almost like around 100 milliseconds after the stimulus onset, we see that there is significant choice stimulus information, which is shown here in red, and that it goes down with time. So we have this so-called primacy profile, if you like. It's largest at the beginning and then declines with time. And then we see that there is also significant choice and intersection information, and that the choice information is very close to the intersection information. Sorry, the intersection information is very close to the choice information. Remember that the intersection information is always upper bounded by the stimulus and the choice information, so it cannot exceed the two, because of course it's a subpart of the stimulus and the choice information. And here is what we see in the other extreme of the processing, which is M1, and for alpha plus beta, we see that there is this ramping of choice information that becomes very strong, and it is significant from, let's say, 0.5 seconds onwards. Now, interestingly, and maybe one of the very novel things that you find here is that there's also significant stimulus information in M1, and that most of this stimulus information is being read out for behavior. So it's a very efficient signal, if you like. Like it contains n bits of information, and about, let's say, 80% of that n bits is read out for behavior. So it's very efficient in that sense. And here I'm showing you in the whole, so the rows are the regions of interest again, and the columns are time. I'm showing you the ratio of the stimulus information to choice information. And what we see is that in the gamma frequency, we have dominantly stimulus information, and in the alpha plus beta, we have dominantly choice information, but in this, which is localized in space, of course, as well. So here, I would like to call this the gamma network, and these ones the alpha plus beta network, which are dissociable, but sometimes they're also overlapping. So this gamma network, for example, we have these parietal cortices, which in the gamma frequency have more stimulus than choice, but in the alpha plus beta have more choice than stimulus. It's very interesting that we have these intermediate regions in the cortex. And here I'm showing just the ratios and claiming that most of the stimulus information in the gamma frequency is choice related, and most of the choice information in the gamma frequency is stimulus related, and most of the stimulus information in the alpha plus beta is choice related. Okay, so far what I have been showing you was the information about the mean or the average of the contrast. So we showed 10 contrasts. We have C1, I don't know, C2 till C10. We get the mean of those at each trial. That would be one number per trial. And then we can compute the mutual information between, let's say, R and S, S being the stimulus, R being the neural response. Now, what you could also do is that you could consider the mutual information between the R and each of the samples that is presented. Let's say C1, C2, C3, up to C10. And what we see nicely is that we see that each of the samples is being presented in V1, in the sense that V1 contains a bump about the first sample and then the second, and if you count these, this will be 10 samples. Each of them is being presented in V1. And again, note that the fidelity or the precision of the encoding is better for the first samples, and it sort of like goes down for the later samples. This is another interesting thing, which I'm not sort of like getting to in this presentation, but if you look at the behavior and if you look at the mutual information between each of these contrasts and the choice, we also see that the first samples in this task affect the behavior more than the later samples. So this like primacy profile is also evident when you look at the behavior of these subjects. Okay, another interesting difference between the encoding of the stimulus in these gamma region, in these gamma networks and the alpha plus beta network, is that I would like to claim that these gamma regions contain information about the instantaneous sample contrast, whereas the alpha plus beta network contain information about the running mean, or the, let's say, like the average of the contrast up to the one that is presented now. So for example, it would be the contrast 1, it would be the mean of contrast 1 and 2 for 2, mean of 1, 2, 3, and so on. So here what I'm showing you is the difference between the stimulus information about the instantaneous sample contrast minus the stimulus information about the accumulated sample contrast. And what we see is that the differences are positive for the gamma regions, for these visual regions, the dorsal visual, the posterior, the MT visual, posterior cingulate, and for these parietal regions as well. And then in the alpha plus beta, we have that the difference is negative for these alpha plus beta regions of the brain, suggesting that they contain more information about the accumulated sample contrast. And that is indeed the decision variable that would be key for doing the task, because they have to make a decision based on the average of these contrasts. So basically what I wanted to claim here is that these two extremes of the processing, they code for different things. So they code for different stimulus features. Next, I'd like to look at the co-information from regions that are from two different hemispheres. So there was a nitty-gritty that I skipped for this presentation, and that was that the information that I showed you so far, they were all computed from the joint responses of the left and the right hemisphere. And so what I am showing you here is a co-information of the responses of the left and right hemisphere about a particular variable, which in this case is a stimulus. And co-information is the difference between the joint information minus some of the information carried independently by each source. And it's well known that if you look at the sign of the co-information, if co-information is positive, then we have more synergy than redundancy. And if it's negative, we have more redundancy than synergy. So it's the difference between the synergy and the redundancy. And note that the way I'm doing, some people do it the other way around and they get negative for synergy and positive for redundancy. I'm doing co-information as the joint minus the sum of the two independent regions. And now we can go and look at the sign of the co-information for the stimulus and for the choice for the different regions of the brain. So here, the rows this time correspond, each row is one anatomical group, and the columns are time. This is the co-information about the stimulus in the gamma, because we knew that gamma frequency mostly contains information about the stimulus. And what we see is that in these like the gamma network, co-information is negative. It means that the left and the right hemisphere contain redundant information about the stimulus. This is kind of what we knew, because these stimuli are presented in the foveal, and then we don't expect lateralization in the visual processing. But it's interesting to sort of like confirm that with these tools that we have. And then we can go and look at the co-information for the choice in the alpha plus beta, because we knew that the alpha plus beta is mostly caring about the choice that the subjects make. But when we look at the sign of the co-information, we see that it's mostly positive. This means that the left and the right hemispheres contain synergistic information about the choice. And this is very interesting. So because most of the studies that look at the synergy and redundancy, they don't look at it in the content base. Here we're saying that there are different patterns of synergy and redundancy, and that the patterns are also dependent on the content that we have. In this case, the stimulus, in this case, the choice. So this is different from the people who look at it from in the resting state, for example. And then here I'm showing you the average for these 10 task-relevant anatomical groups. And what we see is that, interestingly, for these parietal regions, we have significant redundancy in the gamma, and we have significant synergy in the alpha plus beta. So we have this switch of sign, which suggests that they are, as I said before, they're part of these two different networks. Now, you might ask why we get these patterns that we get here. So why would it be that we have more redundancy for visual regions, and why we have more synergy for the motor regions, let's say in the alpha plus beta? And this is a cartoon, this sort of like explanation of why we get these effects. So here are just the neural responses for the left and the right hemisphere in the two different axes. And we have one ellipsoid for example, for the left hemisphere, for one region, and we have another one for the other region. And what we see is that they are in the for the visual regions, the signal correlation is positive and the noise correlation is also positive. So we have they're aligned, and therefore we have more redundancy than synergy. And here, on the contrary, in the motor regions, because the task, as you know, when they respond with the right hand, for example, left cortex is active. When they respond with the left hand, the right motor cortex is active. So the signal correlation would be negative, and the noise correlation is positive because they are structurally connected to each other. And that explains that we have this difference in the sign, and therefore we have more synergy than redundancy. And just so that you know, there's an information breakdown which exactly quantifies what is the contribution of noise correlation to the information carried by these regions. So we could actually go and quantify that by bits as well. Okay, then I look at the onset of information for this task relevant, for a subset of those 21, which we have 10 different groups which are task relevant in either gamma or alpha plus beta. And what I'm showing you here is the stimulus information in the gamma for these different groups. For example, the blue is our visual cortex. This is dorsal visual. This is empty cingulate. This is empty cortex. And this is posterior cingulate, and so on. And then what we can do is that we can just here look at the encoding of the information, and then we can define a threshold based on, let's say, like the values that are before the pre-stimulus values, plus or minus some, plus 3, let's say, standard deviations, and we can compute the onset of information from that for each of these groups. And here's what we see for the gamma. So we see that as we expected, and as we see here, early visual cortex contains the first sort of like, is the earliest region that contains significant stimulus information. And then we have, let's say, dorsal visual, we have posterior cingulate, and later we have the 2 parietal cortical regions. And if you look at the choice information in the alpha plus beta, we see that interestingly, motor cortex and premotor cortex are the 2 regions that contain, that have the earliest onset of information. So information first appears in the motor cortex and then appears in the, for example, parietal cortex or the mid-cingulate cortex and other regions. And this is quite contrary to this like well-known view that information goes to the parietal cortex and then goes to the motor cortex. So we see a different timing here, which is indicative that the information is being propagated in a particular direction here. In the gamma, it suggests that information is first in the early visual and then goes to the downstream regions. In the alpha plus beta, it suggests that choice information is first initiated in the motor regions and then is being propagated into the upstream regions. And what we can do next is that we can use these tools of information theory to actually go and quantify whether information about the stimulus or the choice is being propagated from one region to another. And this is just a cartoon reminder of how these, or a cartoon explanation of how these methods work. You are probably familiar with transfer entropy. Transfer entropy is computes, so we have got this sender X and the receiver, which is Y. It computes the mutual information between the past of the sender and the present of the receiver, conditioned on the past of the receiver, right? So it's telling you like whether, if I condition on the past of the receiver, do I have any mutual information with the past of the sender or not? And this was a directional measure, which sort of like, the idea is that it tells us how much of the information is being transferred from one region to another. Here for the FIT that I explained in the beginning of my talk, we have the feature-specific information transfer, and we have got the past of the sender. Sorry, here actually, yeah, exactly. So we've got the past of the sender and the current and the present of the receiver. So we compute the redundant information, how much information is redundant between the past of the sender and the present of the receiver, and the redundant information which is also unique to the past of the receiver. So because of the PID, we can compute the redundancy and unique information. We have this sort of like this luxury that we could go and compute the amount of the redundant information with the past of the sender, which is unique to the present of the receiver, which is redundant to the past of the sender, but unique to the past of the receiver as well. Okay, then we can go to our task. Here are the 10 anatomical groups. These matrices that I show you, in each of them I've got the 10 sorted by the hierarchy. And here the rows are the gamma frequency, this row is alpha plus beta frequency. We can go and compute the FIT, or the transfer entropy, and then the FIT about the stimulus or the FIT about the choice for each pair of regions. And note that this is directional. All of these are directional. So it's from region 1 to region 2. However, transfer entropy is blind to the content, whereas the FIT is content dependent. It's content specific, actually. So what we see here is that there is some, we can see that the early visual from, let's say like there is significant propagation of information from the early visual cortex to these later regions. And here for the alpha plus beta, by looking at it, we see that this lower triangular matrix is large, suggesting that there is feedback of choice information in the alpha plus beta band. Just something that you might note is that the upper triangular are always connecting one region which is in the earlier in the hierarchy to the later in the hierarchy. So it's always feed forward, and the lower triangles would always be feedback. So then we can go and ask hypotheses about the directionality of information transfer. So what we do is that here again, this row is gamma, this row is alpha plus beta, and for each of the measures, we compute the information propagated in the feed forward direction and the information in the feedback direction. And interestingly, there are two effects that I'd like to highlight here. One is that if you look at the stimulus FIT, there is significantly more feed forward than feedback in the information transfer. This is suggesting that the information goes from the earlier regions to the later regions in the gamma frequency band about the stimulus. And then if you look at the choice FIT, there is significant more feedback than feed forward. So this is suggesting that information about the choice is going in a different frequency band, but from the downstream regions to the upstream regions. And then we can look at the content specificity of information transfer. And when we look at the feed forward connections, we see that in the gamma frequency, there is more transfer of stimulus information than choice information. And in the alpha plus beta, there is, in the feedback direction, there is more transfer of choice information than stimulus information. So this is saying that we have got these two different, let's say, frequency channels, one the gamma, one the alpha plus beta. This channel is sending information in the feed forward direction and about the stimulus. The other channel is sending information in the feedback direction and mostly about the choice. So we saw that these motor areas are the main emitters of choice information and that they contain information stimulus as well. And we saw that they code for the running mean, or which would be the decision variable in this task, of course. So then we ask, how does this stimulus information reach the motor areas? How is it that they contain information about the stimulus? Well, in visual regions, we know that these neurons are selective for the, let's say, the contrast, etc., and the low-level properties. So it makes sense that we see the information in the low-level visual areas. But how does it reach the motor areas? Because we know that, let's say, from 0.5 second onwards, they contain significant stimulus information. So then we asked whether we could address this question with the FIT, that measure that we developed. And then we said that could it be that this information from the gamma band of the visual areas, which is about the instantaneous sample contrast, is being fed to the motor areas, and then they're doing this accumulation and completing the running mean or not. And then what we do is that we do the cross-frequency FIT. So now we're going from gamma to alpha plus beta. And then we compare that to within frequency FIT. So here we're asking, so just let me explain the rows and the columns. So here we've got early visual cortex, for example, dorsal visual, MT visual, posterior cingulate, and superior parietal. These are the regions that contain most of the FIT in the for the stimulus in the feed-forward direction. And here, for each of them, I'm looking at the projections from these regions to the motor region, and then I'm looking at the cross-frequency FIT, so going from gamma to alpha plus beta to within alpha plus beta. So the question we ask is that whether the cross-frequency transfer of information is larger than what we see from the within alpha plus beta. And if it is larger, it's indicative that there is information that is being transferred from the gamma to the alpha plus beta. And what we see here is that in the connection from early visual cortex to motor cortex, there is significantly more cross-frequency FIT than within alpha plus beta. This is only indicative that information is being, that what we saw in the motor areas is probably being, part of it is being transferred from the early visual in the gamma frequency. Okay, now I'd like to recap how the brain supports decision-making in this task based on our data set. So we have, of course, all of our analyses are limited. We look at some data sets and we do some analysis on it. So this is not we have our limitations in our analysis and our design. This task that we had was a visual discrimination task, and we measured activity from the whole brain. Then what we saw is that the information first appeared in these visual areas in the gamma frequency, and that information was about in the gamma band, which is, let's say, 40 to 80 hertz, and that information was about the instantaneous sample contrast, or about the sample contrast that is just present on the screen, if you like. And then we saw that this region also contained choice information and contained intersection information in the same band. So we saw that stimulus information exceeds choice information, and that most of the choice information is stimulus related. And the temporal profile of information showed a primacy effect, which sort of like went down with time, if you like. And then we saw that this stimulus information in the gamma band was transferred in the feed-forward direction to areas that are lower in the hierarchy. So this is, for example, the posterior cingulate, the MT region, dorsal visual MT region, posterior cingulate, and superior parietal cortex. And we saw that that this information goes to this region, which kind of suggests why we see this gamma network that we saw, if you like, in the brain, which they had similar profiles in the gamma frequency. And the region in this, so I wrote them here as well. And then these regions in the gamma network, they all have similar properties to V1, which is larger stimulus and choice information, and primacy profile, and then redundancy of the stimulus information across the left and the right hemisphere. This is another detailed point that I think is very interesting, that when we look at cross hemispheres, we see redundancy of information in all of these gamma networks, if you like, for the stimulus. Much stronger than for what we see for the choice. And then we saw that these motor regions, which are in the sort of like towards the end of the processing hierarchy, they contain a lot of choice information, and that information is being formed in these regions first, and then it's being fed back to the earlier regions. And this goes all the way down to early visual cortex. So it goes to the parietal cortex, it goes to the MT, and also to the early visual cortex, which is the first part of the processing hierarchy. And the regions in this alpha plus beta network, they also have similar properties, which is that they contain larger choice information than stimulus information. They code for the running mean of the sample contrast, which is just the average of the sample contrast up to the one that is being shown now. And the choice information is synergistic across the left and the right hemispheres. And the temporal profile also had this ramping, which is like corresponding to the recency profile, if you like. And then what we saw is that these, I already said this. Okay, and then what we saw is that these, in parallel to the feed forward of the stimulus information in the gamma and feedback of choice information in the alpha plus beta, we saw that there's also information that goes from the early visual to the motor cortex, and from the gamma frequency to the alpha plus beta frequency. So and this kind of explains that this region, the early visual cortex is coding for instantaneous, is sending that signal to the motor cortex, and that is doing the accumulation, if you like, and then computing the choice signal, and then sending that choice signal back to the earlier regions in the brain. So I must say that these results sort of speak against this classic view that information first appears in the visual areas, goes to the parietal LIP, if you like, is accumulated there, and then goes to the motor cortex, and then the choice signal is, and then the subjects make a choice based on the motor activation in that region. So here we're saying that the information is first in the early visual cortex, then goes to the, then appears first in the motor areas, and then that signal is going back to the parietal regions. So this is like, if you see, in direct contrast to this classic view. And the way we do this is by the FIT results that we showed. And also I did some other control analysis that I'm not sort of like showing here, is that, for example, I partialed out the response from the parietal cortex in the motor cortex, and I still get significant choice information there, suggesting that it's not all due to the leakage or any other properties that could come from the parietal cortices. Thank you for your attention.
So you can raise your hands if you have any questions.
Thank you, Dr. Nili,
Sure.
for great presentation. I think the results were brilliant. But I have two questions.
Sure.
You talked about gamma band networks and also alpha plus beta band networks. I want to know that do you see these results in other tasks, or these results, they were shown in this task for the first time? I want to know that can we generalize these networks in other perceptual decision-making tasks? And also, I want to know about other networks, for example, in different resolutions. For example, if we obtain some networks from exactly the same task in, for example, with EEG, can we see these results? And uh, Another question. Um, what is the application of uh, these kind of studies in AI?
Thank you very much. So the first one, which was your first question, was whether this generalizes to the
Yes,
different
yes.
tasks or not. We have done some other tasks. Actually, Tobias Donner's lab, they have done some other tasks which are similar to this, but not quite exactly like this. And this gamma information, this gamma stripe that we saw, and the alpha plus beta, these are pretty much generalizable, I would say. Like this gamma effect is a lot of people see this in their PDM tasks.
I
Also,
have read some
yeah,
of
also the
them.
alpha plus
Yes.
beta, these are pretty common. But then I can only speak against about the data that I have analyzed, which is about this task, these 15 subjects, many trials per subject. But as far as I know, these two effects are pretty much also shown in monkeys as well and humans as well. Yeah. And and about the other question that you asked, whether this could be seen with EEG as well, with fMRI, no, because we don't have a temporal resolution. So with fMRI, we cannot do it. With EEG, I must say that I have done some EEG analysis myself, but I don't believe the spatial localization is as good as MEG. So it's definitely not at the same level. You can work at the sensors, which has some spatial resolution, but first, the leakage problem is very strong there. So for what you get here, it could come from here or there. And then I don't think that people have looked at this with EEG yet. So I don't know the answer to whether we could see the similar thing or not. People don't look at the source signals with this confidence with EEG. But with MEG, we're very we're confident enough to go and look at this for the different regions of the brain. This is with this. If you manage to do source localization with MEG with good resolution, with spatial resolution, it's almost as if you're doing fMRI, but with a very good temporal resolution. So this makes it very suitable for looking at the dynamics and looking at the different regions. So spatial and temporal localization. Yeah. And application for AI. I mean, if you want to make a machine that makes a decision for you, this is telling you how the model would look like, or how one optimal model would look like, which is the brain, and then this is going against the classic view of accumulating and sending it to the motor. But beyond that, I need to think more about that. That's a very good question. Yeah.
Because the human have flexibility in their decision making. Okay, I think these networks in different frequency helps AI to improve. to have flexibility in decision making. Okay, thank
No,
you
I
very
agree.
much.
Yeah, yeah, yeah.
Yes,
Thank you.
Thank you.
Thanks a lot, Dr. Nili. Just as a suggestion, I want to know, can we set this study for basal ganglia and D1 and D2 interaction for this?
Do you mean could we look at basal ganglia and V1 and V2 and how they connected to each other in this talk?
Yes,
Yes, we
absolutely,
could.
because
Yes.
very thanks.
Although there's a problem with the SNR in basal ganglia for MEG. So if you could get a good signal from basal ganglia and V1 and V2 at the same time, yes, nothing stops you from doing that. But whether you can get good signals from these three regions at the same time, that's going to be, I think it's a bit of a tricky case, yeah.
Very good. And can we do this for a specific basal ganglia, for example, accumbens nucleus?
The theory is applicable, but whether you can get good signal, I don't know, yeah.
Oh, okay.
Yeah.
Thanks a lot.
Yeah, these sort of like these regions are pretty tricky to record from. But if you could get someone, or if someone can record from them, yes, this theory is all applicable there as well. Yeah.
Very good. Thanks a lot.
Sure. Thank you.
There's also an online question that I will recite to you.
Sure.
Adele has asked, the stimulus info is in time and frequency dimensions. Is there any chance to calculate the spatial intersection info?
So yes, and I think I showed it to you as well. Let me just go to the slide. So what I said here is that we compute intersection information for time and frequency for all the different cortical regions. And then we can project it to different dimensions. So here I'm looking at for example, how they distributed over frequency. And then here I'm looking at the space and time. So this is intersection information for the gamma and for the alpha plus beta. These are the rows are the different regions of the brain, and the columns are the time. So this shows the spatial-temporal distribution, yes. In general, we have got a 3D tensor, which is time, frequency, and space. And then we're doing statistics on this tensor. So here, again, anything which is gray is non-significant. The parts that are colored are significant. And then we could project it on these different dimensions. One dimension is space and time, which I'm showing here.
Thanks, Ahmed, for your great presentation and interesting findings. As an AI student, I have a question. In your first slides, I think you used SVM. Am I right?
You say SVM?
Yeah, for decoding part.
Definitely, yeah.
Yeah. Because I'm an AI student and I'm working on decoding EEG with deep models. My question is, why in this kind of research works?
Can you speak higher a bit?
I'm sorry. Why is in this kind of research works, it is more common to use traditional models like SVM and some of these linear models, or maybe use a kernel, a non-linear kernel. But I read some newer research works that they use more modern deep neural networks, and they realize the correlation between the representation of the layers in deep neural networks in several layers and the layers in the brain. It's a question for me that why in this kind of research books like decision making, the traditional models are more common.
Because I can tell you, because I sort of come from the same mentality as some of the engineers in this place. If you manage to deliver your point with a simpler method, then that would be much more preferred. Because like all I want to show here is that the the signals or the information profiles, time frequency information profiles from different anatomical groups are distinct from each other. So basically an SVM would do the job for me. But of course I could use more sophisticated methods, and then I need to explain the methods to the audience for 5 minutes and then get to this point. Because SVM is simple, I use that, yeah. Otherwise I could use any other methods as well.
Okay, so you mean that in this stage, because you just want to discriminate some things, the accuracy is not our problems, okay?
No, here the point that I wanted to make was that these different anatomical groups are next to each other. So it is very, very reasonable that we say that, oh, they could be confused with each other a lot. But on the contrary, what I see is that the accuracy is significantly above what I get by chance. This means that the regions from different anatomical groups elicit significantly distinct information profiles. And that's all the claim I wanted to make there. Yeah.
Thank you so much.
Sure.
I believe all the questions are answered. Thank you, Dr. Nili, for your fascinating talk. And let's hear it for Dr. Nili. So according to the schedule, we are now moving on to our poster session. So our executive team members are telling me that before the poster session, we are actually going to take a short break. See you soon in 15 minutes.